Welcome to Arctic Aurora LifeScience Corona Vaccine session. There are so many questions regarding the pandemic and the vaccines and the virus in itself. We are all discussing it and we are all thinking about if we should take the vaccine, whether we will get a vaccine, and when the world can reopen again. And we thought we should collect all knowledge we have in the team and try to answer a lot of those questions that we all bear around us. So my name is Ulrika Slåne-Bjerke and I'm the CIO of the fund. And with me I have Torbjörn Bjerke. He's a medical doctor and he has a vast experience from the pharmaceutical and biotech industry. And we also have Daniel Bolanowski, and he's an expert in biotech. So together we comprise quite a lot of knowledge within the field, and we thought we should try to give you a short overview of what's happening and what can we expect going forward. The first question I would like to ask is, what is the different type of vaccines we are talking about? Daniel and Torbjörn, there are different type of vaccines. We read about mRNA vaccines, for example. So could you explain the different types and what is an mRNA vaccine?
Sure. So to begin with, mRNA vaccines are sort of the most modern vaccines available today. Before COVID-19, there were no approved vaccines of this type. And that is because it's a genetic vaccine. So previously we've been trying to sort of get other ways to activate the immune response towards different diseases, bacteria and virus, by transforming the original infectious bacteria virus in some way. So a lot of lab work has been going on to sort of make those vaccines work and work in a good manner. mRNA vaccines are different because you're dealing with genetic code. So the M in mRNA stands for messenger RNA. Inside our cells, this messenger RNA convenes the message of the genetic code from the DNA inside the cell nucleus outside to into the cells where we have the protein factories. So this message is then translating into proteins. And so we have a tiny illustration of how mRNA vaccines work. So you put these mRNA strings into a lipid layer, which is necessary because if you just inject naked mRNA, it will be destroyed by different enzymes in your body. So you need some sort of protection and also help this mRNA to get into cells.
So can you explain lipid layer get into the body?
Yeah.
What is it actually you put into the body?
So it's different fats, different compositions. They've been working quite a lot, these vaccine companies and other companies also that are trying to put proteins and DNA into cells in order to have a good transport vehicle.
So this is fat?
Yeah, and actually it resembles sort of our cell membrane structure a little bit. So when you make these sort of bubbles, they form sort of very nice spherical particles, and then you can put whatever you want inside there, and it's protected from the sort of environment in your blood, for example.
So the genetic code for the vaccine is put into fat and then put into our body.
Exactly.
And the fat layer makes it so easy for this droplet to get into the cells.
Yeah. And
that should work.
That's essential. Yeah.
Because our cell wall likes to open up for fat.
Yeah, it's lipophilic.
Yeah.
So instead of the information coming from our DNA translated into mRNA, then it's translated into protein, you sort of circumvent DNA out of the equation and just insert mRNA into cells, and then we can manufacture whatever we want. So this could have promise in the future of sort of in how we take medicines, because this mRNA could be used to make the cells manufacture pretty much anything. However, when it comes to drugs, you need a lot of amount of protein, right? And that's been the limitation of mRNA in that applications. Vaccines, however, you don't need that much of it. So you just need something that triggers the immune response. So in this case, what this mRNA code contains with regards to the COVID-19 vaccine is the code for the cells to produce the so-called spike protein that surrounds the coronavirus. We've all seen sort of images of the coronavirus, and actually the spike protein is the reason why it's called coronavirus in the first place. If you look through it in an electron microscope, it looks like they have a sort of a crown, corona in Spanish, I believe. So by making that protein, by injecting this mRNA, the cells start producing this protein. The proteins are then moving out of the cells into, let's say, the bloodstream, and then it's recognized as a foreign particle by the immune system.
One question that I have got regarding this is, does this affect our genetic code in general?
No.
No.
So mRNA does not integrate with DNA, so it's a completely different entity. It's an interesting question because we will get to that. There's DNA vaccines as well, where you inject DNA instead of RNA. So that perhaps a little bit more sort of a more important question there. But in this setting, the answer is no. It does not interfere with our own genetic code. No.
So to summarize, you have the mRNA vaccines, corona vaccines, and then you have the traditional type where you have like a kind vaccine type of vector.
Yeah, it's a middle thing, and that's the so mRNA vaccine manufacturers today are Moderna and BioNTech, that is partnered with Pfizer. Those we know that they have data. We'll get to that as well, how they have worked. We have a couple of other players like CureVac that are also have mRNA vaccines in development, and then there are companies like AstraZeneca and Johnson & Johnson. They have sort of a similar approach in that you're trying to put genetic code into cells, but you do it slightly different. Now we're talking DNA, and we're talking actually other types of viruses. In this case, a class of viruses called adeno-associated viruses. In sort of their natural origin, they may give us humans colds, like really mild colds. But what you do is you take this virus and then you modify it. So you take away all the sort of pathogenic properties, the stuff that makes you sick. For example, these viruses cannot replicate within cells. The only thing they do, and this is what viruses do very well, is inject DNA into host cells. So back to your question, potentially this injection could actually integrate with our own DNA. With adeno-associated virus, that does not happen. And we've seen that in other settings. Adeno-associated viruses is used in gene therapy, for example. And the only thing they do is inject a piece of DNA into the cell, but it does not integrate into the whole genome.
Is this similar to flu vaccines?
The flu vaccines there are different kinds of flu vaccines.
Okay.
So you
can't really compare.
No, but flu vaccines.
Comparative principles.
In principle, yes, but flu vaccines traditionally have been other means of or other types of vaccines. So besides the adeno-associated viruses and the mRNAs, there are more sort of traditional approaches of making vaccines. One such example is that you take your pathogenic virus. And then you multiply it a lot of times in the lab. And what happens is, we'll get to mutations as well, but these viruses change, and if you do this many times, the virus will actually become less and less disease-causing. It will still be active, but it's a very weak virus. So this is called inactivated viruses. And that's been used in other types of diseases, like measles, for example. The vaccines they give there is a very weak measles vaccine that does not give cause to the typical symptoms, but it is enough to get you an immune response. So that's one type. And then there are sort of hybrids where you can take one virus, for example, the measles virus, and this is actually what Merck tried to do in COVID-19. Took one of these very weak measles viruses and then put the spike protein onto it. So the immune system is supposed to recognize the spike protein. So the next time the COVID-19 virus enters the body, then the immune system goes, aha, I will attack you. This approach did not work very well. We don't know exactly why. It has worked in other diseases. But in this particular instance, Merck decided to shut that down.
Okay, so so the picture is we have mRNA vaccines,
yes,
and we have the adenovirus.
Yes.
vaccines.
And then the sort of weakened or attenuated vaccines, as we call them. And then in addition, we have the pure DNA vaccines. And they work similarly to the mRNA vaccines, in that you're injecting sort of naked mRNA or DNA with the help of these lipids or fats. We have yet to see any clinical data from the DNA vaccines, but they're under development.
So, so there are two main type of vaccines currently available, and there are other under development. So, what about how well do those vaccines protect? I mean, how can you tell if they are effective or not?
Sure. So as an example, we'll take the Moderna phase 3 study. So that will serve as a good explanation for how you sort of discover whether these vaccines are active or not. So you take a number of people in the range of, in this case, to be really sure what you're talking about, you need a big statistical supply. So let's say you take 30,000 people, healthy volunteers in different ages, because you want to see how this works in different ages. And then you either inject them with this vaccine candidate or with a placebo. So the people in this study do not know whether they have the active vaccine or not. And then you just let them out in society and then check and see if these people
go by bus and train.
Exactly. And then see if these people get infected with the disease, or actually if they get symptoms, because it's very difficult to test these people every day to see if they have a molecular trace of the virus or not. But if you have symptoms, then that's probably enough. And this is what Moderna did. And so you soon discover how many people in the placebo arm of the study and how many people in the vaccinated arm of the study get the disease. And in Moderna's case, it was pretty overwhelming. I think we have a graph of that, a so-called Kaplan-Meier graph, where you see on one hand the placebo group. For every step up in this graph, you have one person getting symptomatic COVID-19 disease. And then you can see how flat the vaccinated curve is, or the vaccinated line.
Fantastic.
And that's a major difference between the two arms, obviously. And then, so what Moderna said is that they have approximately, I think, 94% vaccine efficacy. So what that means, if you have a sample of, say, 100 people, again, half of them, you make a sample from this study of 100 people, that get infected by the vaccine, infected by COVID, and then see how many that took the active vaccine, how many took placebo. In this case, 94 people were on placebo, and only 6 people got the vaccine. That's how these efficacy numbers are calculated.
Highly efficacious.
Highly efficacious, yes.
So we are protected if we are vaccinated. So, but then also reading newspapers and listening to the news, there are now mutations that causes variants of the coronavirus. And how does that affect the efficacy of the existing vaccines available?
So we have 2 examples of this, 2 sort of mutational strains. One is the so-called UK strain, one is the so-called South African strain. And only 2 companies were late enough in development to include people that might be affected by these 2 different strains. Pfizer and Moderna studies were too early, so they had the original COVID-19 strain, whereas to begin with, Novavax made a clinical study in the UK, and approximately half of the people that ended up getting infected in that study, placebo or not, or with the active vaccine, had the so-called UK mutational strain. And they got an 86% protection versus 96% protection on the original strain. So I'd say that's a pretty good result.
Yeah,
it's a weakening of the immune response from that data, but it's still pretty good. On the other hand, the South African vaccine is a little more dire, and we should be a little caution there, and other mutations as well. So the corresponding data there was about 60%. A little bit difficult in South Africa because you have HIV, a large population with HIV positive, and they have weakened immune system. But in those that are HIV negative, you got 60% protection.
but
So considerably lower.
Yeah.
And you're on the sort of threshold of what you can call an efficacious vaccine. It's still efficacious, and if you were to vaccinate a whole population with 60% efficacy, you'd still probably reach herd immunity after a while.
Yeah, absolutely. And also in terms of the Pfizer BioNTech vaccine, they tested the new variants with antibodies from previous vaccinated subjects, and could actually neutralize the virus.
Yeah. Again, you saw a numerical difference. It was slightly less efficacious, but still pretty good.
Yeah.
And also, even though you might get the viral infection, you do not in the same extent get serious disease. Isn't that so?
No, yeah, that has been the case with all tested vaccinations so far. That you don't get really sick, but you get a mild disease. But we must not make any mistakes. There will come a mutation that do not, where the existing vaccines do not really get a good immune response. But here we have the mRNA vaccines, and we have the others that can quickly come into play in development. So we could have a situation like we have with the flu, that maybe every second year we need to be vaccinated or boosted.
Yeah. So here, this is a big difference between the traditional vaccines. I mentioned how you sort of, in theory, manufacture these attenuated viruses. You need to work with them in the lab for quite a long time, and making a vaccine that works for a new mutation could be pretty tiresome. Whereas with mRNA, you just sequence the new virus, and then you have your mRNA vaccine sort of the next day, in theory.
Yeah.
So easily explained, you change the code and you put it in fat again, and then in the body, and then you get protection from the new variant of the virus by the changed code. So these vaccines, how do you manufacture? How do you produce vaccines?
Yeah. So with mRNA, that's potentially an advantage as well, because with a lot of sort of protein drugs, for example, you need some sort of a living thing to produce these proteins. Bacteria, a lot of the influenza vaccines are made through chicken eggs, which is in itself pretty sort of cumbersome, especially if you need high amounts of them. mRNA can be manufactured completely synthetically. So you just put the different letters of the genetic code into your vaccine, and that's pretty much how it's done. Now, we've all heard of sort of the supply shortages right now with all vaccines, because it still takes a bit of time to manufacture. You need to validate all the steps, etc. It's not a super easy thing to do, but in theory, once we have all this infrastructure in place, then you can make a lot of mRNA vaccines pretty quickly.
To me, it sounds like rocket science. Anyway, you manufacture a genetic code synthetically and then put it into fat and into the body. I mean, it's still super innovative, I would say.
Yes. Problem is, and I guess we'll get to that, is besides ramping up production, is how you then distribute these vaccines, and that's for now the main drawback of the mRNA vaccines.
So do all mRNA vaccines require minus 80 degrees?
For now, we know that the Pfizer vaccine does. The Moderna vaccine only requires a minus 20 freezing temperature, which is sort of household freezer. So that sort of simplifies things a little bit, but it's still low temperatures. Of course, minus 70, which is the requirement for Pfizer's, is an infrastructure challenge. I think we can do fairly well in sort of Western countries. But remains to be
still difficult out there in the small cities.
So, for example, one thing you can do with traditional viruses is to send them out to small clinics, your local clinics, or even if you go into sort of elderly care, you can have someone just bringing the vaccine there, and that's not possible with these mRNA vaccines. So probably, or what we see now is a lot of countries are setting up huge vaccination facilities in sports stadiums and the likes, because you need a centralized freezing supply in order to make the vaccine not expire.
But Pfizer BioNTech will also come as a second generation coming soon. I think will solve that problem with the minus 80 degrees and go higher up.
It's probable.
So, They have that in the pipeline.
Okay, so next part will be dosing. How are those vaccine dosed, and why are we taking 2 doses as it is for most vaccines?
Yes, so we need, as Daniel alluded to, we need to activate the immune system. And when we are getting infected by the virus, if we look at that in the first place, then the virus comes in, it destroys the cells, it goes into the cells and destroys them, and at the same time they also activate the immune system to produce the antibodies that will eventually kill the virus. But that is, first of all, they destroy a lot of cells, both in the upper airways and in the lower airways. With the vaccination, with the mRNA, we insert a new protein that is not infectious. So it doesn't destroy cells, but it activates the immune system as the second step also for the virus, as I said. And that is extremely important because it activates both so-called B cells, going to plasma cells, producing the antibodies, but also T cells that are important cells in the fight against the virus is being activated. And that's also why we are measuring 2 types of immunity after a vaccination, after the virus infection. That is the antibody response that is produced by the B and plasma cells, and then the T cell response, which is also a bit delayed, but very important part of the immune system to fight the virus. So in the first place, in the first dose, you get the immune system activated. And it starts to produce the antibodies, it starts to activate the T cells. But it has been shown, especially for mRNA, that you need a booster, you need the second dose, about 21 days, they have measured 21 days after the first dose, in order to get the full protection, as Daniel were talking about. So then when the second dose comes in, then there's much more activities in the immune system, much more antibodies being produced, and then many more T cells being activated, because now the immune system recognizes the virus or the dead virus, the spike proteins, and that's why we need 2 doses.
So if the second dose is delayed, what happens? What do we know about how long time span we can have?
We don't know for sure, but it's extremely important, as a statement that you get back and you take your second dose. You do not skip your second dose. That's very important. So you need to have both doses in order to have a full protection.
So Torbjörn, from the immune response perspective, having a high immune response is sort of a quantitative issue. When you have an actual infection, COVID-19, you have massive amounts of virus. So you also have a massive immune response. That's actually part of the problem why some develop very severe symptoms, because the immune response can run off and go crazy, right?
Yes.
But then at least you have an immune response.
Yeah.
With mRNA and other vaccines, the point is that you're only injecting sort of a minute amount of this protein, and as soon as the antibodies start recognizing them, then you have a very quick sort of killing of these particles, and then you're done with, and that's not a real immune response. It's just a very, very quick one.
Yeah, but it's a real specific immune response. Yeah. So you don't, as you say, with the real virus, you kill cells and you activate in a very bad way the effector cells in the immune system. For example, the so-called macrophages, they become killer cells. They produce a lot of cytokines that just get the inflammation to go totally crazy. You don't get that with the vaccination. There you get a very specific, very intelligent immune response that protects you.
So Johnson & Johnson has a vaccine candidate where 1 dose might be sufficient. So what does that have? What type of implications does that have, and where do you see that in the treatment or in the vaccination of populations?
I would have no problem to take the Johnson & Johnson vaccine. It's one shot, and you get about 70% protection compared to 97 or 98 or whatever in that range. And that can be distributed in a fridge, so it's easy to be distributed around the world. So it has really its advantages.
So do we believe that the data shows that it's enough efficacy?
It is enough efficacy, but as a kind of an immunologist, I would say it would be good with maybe another booster, and we come back to that because in the best of the world, we get our vaccination now, and then we get a booster every year, or every second year, in order to keep the immune system alert towards the virus. And then, of course, we'll monitor all the variants coming up. And if they go totally crazy, we need a new vaccination. We know that, and the companies are already in action doing new vaccinations on those new variants. But I believe that a certain day there will be a variant that doesn't really fit into the vaccination we have nowadays, and then we need to do it again. The good thing is that we know how to do it.
Yep.
The companies know how to do it.
The key here is sort of short-term, medium-term, and long-term. And short-term is sort of days and weeks, and we know from the mRNA vaccines that you need to take this booster shot.
Yeah.
We know in the short term you don't need to take it with J&J's vaccine, which is a big plus for that. And long term, we don't know anything at all, really. What we can expect from other vaccines is we're talking a year, perhaps, maybe two. And then perhaps we need to sort of refresh our immune system with another vaccine.
So I like to bring us forward into us as individuals. For us, how should we relate to the vaccine? I mean, is it safe to take the vaccine? You say you don't know anything about the long-term effects. How safe are those?
From what we know today, and these, as you said, Daniel, these are trials with more than 30,000 subjects in. We don't see a lot of side effects. We see some local side effects on where you get the shot, but it's too early to say if there are any larger side effects in there. Then you need to take that into consideration in terms of herd immunity, because we need herd immunity on this one. This is not a normal flu. This one kills a lot more people than a normal flu. So we need the herd immunity in order for a society to work again and function normally. I would say as a doctor, when I do the positive and the minuses on this, I would definitely say there are many more pluses than minuses.
So how will a protection? How long will it last?
Yeah, that's a fantastic question because we don't know. As I said, I believe that we need to be boosted every year, maybe, or every second year, and then we need to be revaccinated with the new strains. This is the COVID-19 is here to stay, so we need to get used to it and how to protect ourselves in the best way.
And I wonder if I start with getting vaccine A, and then I take vaccine B.
Yeah.
So what will happen then?
Oh, then this fantastic, beautiful immune system will then recognize the other one, and you will have some spillover, but there are some new antigens in that new vaccines as well that will be boosted in this new vaccine. So you will have both a reactivation of the older one if they combine for example, the spike proteins, different parts of the spike proteins, and then you will have a new vaccination of the new variants. So it will build immunity in your own body.
So it can actually be an advantage of having one vaccine and then I get another type of vaccine.
Yes.
and you can kind of build your protection.
Yes, yes. We don't know when this virus will stop mutating, or when it will. You know, the new strains that we have talked about. It seems that they are much better in getting the subjects sick, right? Because there are more viruses coming in. We know now quite a bit on how the virus binds to the cells and what binding factor is important. And it seems that this is enhanced in the new strains.
Yeah.
Maybe next time there are some other features of the new strains that needs to be taken care of. But I'm quite sure that our technologies that has been developed during the last year will also take care of that. So yes, you can.
I think the good thing with coronavirus is that I think the thing that could potentially make this difficult, and what we've seen so far, and what we've seen from the differences from the original SARS virus to the SARS-2, which is the COVID-19 causing virus, is that the spike protein is key. And we're also seeing with the mutations that perhaps make the disease more transmissible, is that these strains have a better, from the virus point of view, the spike protein has improved, so it binds more easily to the host cells.
Yeah, that was what I said.
Yes, yeah.
But in a different way.
Yes.
So if I am vaccinated,
Yeah.
will it be okay to visit my old parents, or
No,
how safe am I from contaminating others?
Yeah, and this is extremely important, because when you get your first vaccination, it takes at least 2 weeks before the immune system gets to produce what it should produce, and that's the antibodies and the T cell response. So you need to be protected. You need to keep the distance and do what we are doing now for at least 2 weeks' time, and probably longer time. So I would say, if you're getting the 2 shots, you need to be totally protected in those 21 days at least.
And you probably need to monitor how, from a societal perspective, how this vaccine is, this virus is spreading. If we still have a lot of spread in the society, and you have, let's say, a 90% efficacy of the vaccine, well, then there's a 10% chance that you will actually get COVID-19 still, and then you can probably have other people get sick because of you. So that's, I mean, if we're at a stage where we push down the number of affected people as a whole in society, then it's probably much more safer.
And that's why herd immunity is so important. And by the way, when you are vaccinated, you can still transmit the virus, because you can be in contact with a COVID-19 patient, or you can put your hands on a glass that has COVID-19 on, and then you can transmit it.
And also,
so it's important, one, the herd immunity, but it's also important for quite some time to take care of and think about what
you're still sensitive.
You're still sensitive, and you'll be that for.
And from we know for the immune system, even if you're vaccinated, even if you had the disease already, if you had a COVID-19 infection, you can get infected again, and you may have the virus to grow inside your cells for at least a couple of days before your immune system really kicks off. During that time, you can easily make other people sick as well.
It's an important point, Daniel, because in the first place, we thought, okay, have you had COVID-19? You're on this top league in the society, and you can't get it again. But we see that so many times that actually you can be reinfected. You don't get the full symptoms, but you get some symptoms, right? You get the mild symptoms. So it's a lot that we don't know yet about the virus and how it transmits and how.
But actually, if we can come back to a previous point, is that we're talking about herd immunity, perhaps here in Sweden, in Norway, in the US. That's one thing. But unless we develop herd immunity globally, we will have these mutations going wild. If a lot of people are not being vaccinated in Africa, we're already seeing the South African mutation taking a grip on that continent. If they're not vaccinated, then it will probably continue to spread, and we'll have another mutations that will probably make our vaccines that we have today not very useful.
Yeah. So it's
that's the importance of a lot of vaccines being available now in the sort of pandemic state, so we can suppress this.
Yeah, I plan to wrap up a bit about vaccines, efficacy, us as individuals, and then go on to the companies and the stocks and the appreciation in the stock market. But is there anything you'd like to add in terms of how they are working, what should one do? Final remark.
We could be talking about this for at least 3 hours more, but I think we should go to your...
Yeah, I just want one more thing, and that's what we touched upon, safety, and this is an important thing because a lot of people are doubting whether they should take the vaccine or not. Again, we don't have super long-term exposure to this. We have, I guess, the first phase 1 patients were sort of are almost 1 year into their first shots, so we will have some sort of indication there. What we can see from a technical point of view, the mRNA vaccines are really simple. It's 2 components. It's the different lipids, and you have the mRNA strain. The mRNA strain will not cause any. I very much doubt that it will cause any adverse events. Potentially, these lipids will maybe, in a sort of a theoretical space, cause something, but they are broken down pretty quickly. They're not disease spreading. You can't get an infectious disease out of these. So potentially, I think, or from a technical point of view, it should be safe.
And I will comment on this later, but it's not that this is something that's invented in March this year. This is something that companies have been working on for 2 decades at least. So the technology was there, but it hadn't really gone into commercial applications or medical applications. So the safety work has been done during a long time, or many years.
Moderna is sort of the trailblazer in mRNA vaccines, and they have been trying. They have clinical trials in other types of viruses before the COVID-19 pandemic, and have not seen any long-term issues. So just to repeat, what we do see when you get the vaccine is you can get in sort of an injection site reaction, right? It can hurt for a couple of days. Mild fever, a transient fever, a couple of days is common. Headache, stuff like that. Usually a couple of days. What some people may have reacted to are more exotic things that occurs in these studies. And what you should keep in mind is that we're having hundreds of thousands of people now in clinical trials for vaccines. The odds of hundreds of thousands of people, if you have one case of someone getting a cancer diagnosis, there was an early report of a multiple sclerosis MS diagnosis. Those things will happen in a population, so you need to separate that from what the vaccine might be doing, and those that are running these trials are looking very carefully at all of these different things and trying their best to judge, could this possibly be vaccine related, and we have not seen any severe issues. The only, I guess, thing that you should be cautious about, and the people that are giving these vaccines are very well aware of that, is you can get an allergic reaction.
But
to
you
the
can
vaccine.
Get that with any vaccine.
Any vaccine.
Any medication.
Yes, and we've seen that people that are already allergic to something, nuts, fruit, something, have a higher propensity to get an allergic reaction.
That's why you need to wait for 15 minutes after you get the shot.
Yeah.
If something happens there, then you need to be treated. If not, you can go home.
Yeah, but that's, from what we just said, that's the only thing that we've observed so far with these vaccines. So I think overall, especially if you compare to the alternative, not getting vaccinated and potentially getting sick with COVID-19, that's far worse.
So we will finish up with a few points from a fund manager's perspective. And if you look at those specialized mRNA companies, for example, they have had fantastic stock price appreciations during the year. So Moderna's stock price is up 687% in 1 year. BioNTech is up 290%, and Novavax is up more than 3,600% the last year. And of course, this has been positive for the return of our fund. And the question is, of course, what do we expect going forward? So from the vaccine companies and also from the large pharmas that are involved in vaccine development now, we expect huge sales numbers in 21 and 22. And what we also realized due to the mutations and the variations is that they will probably be able to sell their vaccines on a more regular maintenance basis going forward. This is, of course, creating a lot of value for society, and it's also creating a lot of value for shareholders. And we, as a fund, we focus on when innovation meets medical need. And this is a clear example when the biotech and the pharma industries solved, or at least is on the way of solving the problem globally for the world. And there are so many other diseases and therapeutic areas where you have huge unmet medical needs. And our companies involved in the industry is working on solving those problems. And those companies, for us, will be investment opportunities for our funds. So therefore, going forward, we still have a very positive view on the sector. As I mentioned before, the platform and the knowledge that those vaccines are based on has been worked on for at least 2 decades. And these technologies can now be applied in other therapeutic areas. So we are looking forward to a continued transformation of the way you manufacture and develop and produce and market new products in the biopharmaceutical sector. So with that said, I and we hope that you have more information regarding vaccines and that you have found this session interesting. Thank you so much for listening.